Okay, so welcome back for the afternoon session. Our first speaker is Drew Myerscough. He's an undergrad at Rice University in Joff Silberg's lab. He's majoring in biochemistry and minoring in cell biology. And he's worked with Joff on a number of different projects, including using directed evolution to improve halide recognition by a methyl halide transferase reporter, which he won an NSF poster award for. And he's one of the authors on a recent study probing the structure of an electron transport chain in a multiheme protein that's involved in extracellular electron transfer. And today he's going to talk to us about determinants of multiheme cytochrome electron transfer uncovered by systemic mutation. Okay, Drew, go for it.
Yep. So I'll be talking about a project that I worked on with my mentor, Dr. Ian Campbell, in which we systematically mutated a multiheme cytochrome called MtrA, which electroactive microbes in the genus Schumanella use to conduct electrons from intracellular catabolic processes to extracellular materials, specifically like metals and minerals. So for a little bit of background, in the Silberg lab, we're really interested in understanding sequence structure, electron transfer relationships, and natural proteins. Previously, we've done a lot of work on like the 2-iron-2 sulfur ferredoxins and 4-iron-4 sulfur ferredoxins, including with like Vic's group and so on. Recently, though, we started working with flavodoxins and cytochromes. And a recent study by a postdoc in our group, Josh Atkinson, did some really interesting engineering work with the MTR complex from Schumanella in E. coli, where he basically used a variety of different electron transfer proteins from different species to kind of rewire the electron fluxome to create a living bioelectronic sensor. And shown here, performance under ideal lab conditions, sensing 4-hydroxytamoxifen, which is just like a common like inducer in like antibiotic or like estrogen mimic actually. So then shown here is performance in real world samples taken from rivers and beaches in Texas. And it actually performs surprisingly well, and it's able to monitor the concentrations of these contaminants in real time, which is pretty cool. And for this study, in addition to just other fundamental and applied studies, we tend to use growth selections, which allow us to couple the function of a particular electron transfer protein to cell growth. And shown here is an example, older figure from ferredoxin studies that we've done in the past, where basically a cell is only able to grow if it is expressing a functional protein electron carrier that is able to conduct electrons from a donor to an acceptor. And only the cells that are expressing a functional ferredoxin in this case will be able to multiply. And if you sequence the resulting culture, then you'll be able to see which proteins are the most active. And a typical workflow will look something like this, where you generate a DNA library encoding a variety of mutants. These can be like point mutants where you change one amino acid to another, or they can be insertion mutants, which I'll be talking about today, where you insert peptides at different places in a protein. You could also generate chimeras by combining different proteins in a particular family. And you can even just take a bunch of proteins from different species and express them all together if you want, assuming that you can synthesize them small enough. But once you have your library, you can transform it into some sort of mutant that is only able to grow if it is expressing a functional version of that protein, and enrich for those functional mutants using a growth selection with respective control, non-selective. And by sequencing the DNA extracts from these cultures, you can generate a mutational profile based on the relative enrichments of the different mutants in the library. So today I'm going to be talking about one of these projects, specifically with the MTR-CAB complex from Schumanella onadensis. Shown here is the crystal structure of MTR from Schumanella baltica. And MtrA, as you can kind of see here, is like it's kind of like a protein nanowire. It's like a decaheme cytochrome that takes electrons from internal metabolic processes and then shuttles them through the membrane to MtrC, which interacts with extracellular materials. I don't know if like any of you have seen Joff give versions of this kind of presentation, but he really likes to include pictures of his dog Stormy. So I'm going to do it my way, and I include my cat O'Shea. And like the rest of us, she has a preferred nutrient source, which she breaks down into acetyl-CoA and the TCA cycle intermediates that we know and love. And as they're oxidized, the electrons are conducted to oxygen, which is reduced to water. Schuenella does things a little bit differently. It really likes lactic acid. And when it oxidizes it, it conducts the electrons to minerals, and also just metals and soluble electron acceptors. And these can include a wide variety of transition metals, as well as some exotic stuff like uranium. In addition to bulk minerals like hematite, metal oxide nanoparticles, as well as organic small molecules and metal electrodes. So to do this, most Schuenella species have a wide variety of MtrA paralogs. Shown here is a really nice review by Jeff Gralnick's lab at the University of Minnesota, describing the variety of different paralogs that are present in Schuenella omedensis. Usually they're kind of optimized for particular metabolites, but they're also pretty widely interchangeable, so it's a very modular metabolic setup. And Jeff Gralnick's lab has generated a bunch of Schuenella mutants where these paralogs of MtrA have been knocked out, as well as some other small paraplasmic cytochromes that are involved in other metabolic pathways. And what's really nice about these strains is that they display different oxytrophies. So, for example, strain JG665, which we used throughout this study, requires addition of functional MtrA for growth on ferric citrate under anaerobic conditions. So if there's like lactate or some like oxidizable carbon source in the medium, Schuenella cannot put the electrons from it anywhere if it does not have MtrA, or there's some alternative electron acceptor. So using this strain setup, our goal was to use a growth selection to evaluate the functionality of all MtrA insertion mutants with a particular peptide insertion. And our workflow for this is to generate a library of all MtrA mutants with a short peptide inserted after every position in the protein. Then grow up bacteria that are expressing these MtrA mutants under selective and non-selective conditions. Use deep sequencing to compare the relative enrichments of the mutants. Characterize some interesting mutants in a little bit more biochemical detail. And then look at the MTR structure to see if we can identify any determinants of mutational tolerance. So to generate the library of MTRA mutants, we used an algorithm called SPINE, which was reported in the literature a couple of years ago. And it's a really nice alternative to transposon-mediated mutagenesis because it's a lot less biased. All of the oligos are made synthetically. And traditionally, this has been a bit of a problem for longer proteins because it's pretty expensive to synthesize long DNA sequences. So the way that it works is you break up the coding sequence of a gene into tiles that you then generate synthetic oligos for that contain DNA encoding the insertion of interest after every codon in the native protein. And after synthesizing a pool of these oligos, you can clone them into the respective tile positions in your original plasmid. And you eventually end up with a library where every mutant has exactly one insertion, and there is hopefully coverage for every possible insertion variant. So after generating this library, we grew up the cells expressed in this library in a naive culture, so non-selective conditions, and then we transferred them to a fumarate culture because this strain does not require functional MtrA to grow on fumarate. This is kind of our control non-selective. And cells in selective conditions growing in ferric citrate under anaerobic conditions. And these are the deep sequencing reads. You can see for the fumarate conditions, as you'd hope, the relative abundance of each mutant is pretty even. There's a little dip here. This was also present in the naive library, so it seems to have been an issue in the DNA synthesis, which just happens sometimes. But when you look at the selective conditions, it's a lot more interesting. There's a lot of variation in the enrichment of different mutants, particularly like pockets towards the Determinants. And then you also see generally most positions are pretty comparably enriched for the rest of the protein, except for these very steep basins that occur periodically along the sequence. And when we map these differences onto the structure of the MtrA, what we see is that they line up very well with the heme insertion sites. Included in these annotations below are a couple of other functional domains, such as the interface with MTRB. For the most part, the MTRB interface seems to be relatively tolerant. There's an area right here that has a pretty low insertion tolerance, but because it's between two hemes, it's kind of hard to tell if that is a result of disruption of electron flow or if it's a disruption of interactions with MTRB. Surprisingly, the MTRC interface was actually very tolerant. And in fact, the insertion that's most enriched in the library, which I'll talk about a little bit more later, is actually in the MTRC binding interface. Shown below are the 10 heme binding motifs, which are 2 cysteines with 2 amino acids between them, and a histidine that kind of coordinates the iron atom in the heme. So based on this distribution, we picked a few mutants that would be kind of interesting to look at further. We picked a couple in the N-terminus. So there's a signal peptide at the N-terminus of MtrA that's responsible for translocation into the periplasmic space, as well as an N-terminal disorder domain that mediates interactions with periplasmic cytochromes so that MtrA can accept electrons from different electron hubs that kind of gather electrons from catabolic processes. I won't be talking about those very much today because it's a little intensive and you gotta keep the talk kind of short. But we picked 3 insertions that are in the heme binding motifs and 3 that are just kind of distributed throughout like the Z-score distribution. So like this insertion 158 is near 0, so it's about average. This alanine insertion is like slightly enriched, but not as much as insertion 301, which is the one that's in the MtrC interface, and this is the most enriched in the library. And then going back to the like Z-score versus like across MtrA, these are the like relative positions of each of the mutants. And first, we cloned each of the mutants out of the library and grew them up on ferric citrate under selective conditions just to make sure that they behaved the same way on their own as they did in the library. And what we see is after 24 hours, all of the insertion variants except for 261, which is within one of the heme binding motifs, are enriched to about the same degree as wild type, which is pretty surprising. In a lot of like soluble enzymes and stuff, an insertion of this size will like pretty drastically disrupt enzyme function or stability. But MtrA seems to be pretty robust to that. And even more surprising is after 48 hours, this insertion within a heme binding motif grew to about the same final density as the wild type and the other insertion variants. So the takeaway was that even the least enriched MtrA mutants are still pretty functional. And after that, we decided to clone a few more insertion variants that were within the heme binding motifs. The A's here just mean like the alpha helices that coordinate the hemes. There's 10 of them, 1 for each heme. And we picked out 3 of them. And again, after 24 hours, they're enriched to varying degrees, but not a whole lot compared to wild type. But after 48 hours, they all reach about the same endpoint. And given that these are like pretty drastic disruptions of MtrA structure, we think that this indicates that insertions that disrupt the heme binding motif still support electron transfer. So here it just kind of depends on what's rate limiting in the metabolism of the organism and how much the loss of a particular heme is going to interrupt electron flow. To characterize these mutants in a little bit more detail, we performed some chronoamperometry studies with the Otto Franklin lab at Rice. This is a pretty low throughput assay, so we were only able to characterize a few mutants. We picked one mutant that was in a heme binding motif, 103 here, as well as a couple that were just kind of throughout the distribution, like this like 205, as well as, again, insertion 301, which is the most enriched. And what we see here is just kind of like the current that the cells are, the current that the cells are generating versus time as they grow under anaerobic conditions with lactate as a carbon source. And if you integrate over time, you get the total charge that these cells have conveyed to the electrodes. And the results that we see pretty closely match the results that we saw for the growth selection on ferric citrate, where insertion 103, which is within one of the heme binding motifs, is not really generating as much current, which is consistent with the loss of a heme, or just like a general disruption of three-dimensional structure. Insertion 261, which is also like a relatively low enrichment insertion, is also pretty depleted. But insertion 301, which was the most enriched in the library, is growing pretty comparable to wild type. And although there's only a few data points, if we plot these relative to the z-scored enrichment on ferric citrate, the total charge generated with the insoluble electrode as an electron receptor correlates pretty well with the performance of these mutants on citrate as a soluble electron acceptor, which is also pretty interesting and somewhat unexpected. There's still some debate on this in the literature, but there is some evidence that MtrA can actually reduce citrate and, to a lesser extent, electrodes in the absence of MtrC. So this might reflect that and could also potentially be related to why the MtrC interface is so like robust to peptide insertion. So after doing these biochemical studies, we decided to take a step back and look at enrichment across the protein as a whole and see if we could find any structural features of MtrA and the MtrCAB complex as a whole that correlated well with insertion tolerance. For a few, we looked at like the radial position of the mutation relative to the central axis of iron atoms in the hemes, as well as kind of the axial position along that long axis. So basically where the insertion site is relative to the periplasmic versus extracellular kind of axis, as well as crystallographic B factor, which is commonly used as a proxy for flexibility, in addition to intraprotein and interprotein contacts, which kind of incorporate both structural and functional aspects. The intraprotein contact density kind of indicates like the volume available to accommodate an insertion, whereas the interprotein contacts get at like the number of interactions that are possible with MtrC and MtrA, or MtrC and MtrB. And I also did a few others, but can't really talk about them, so I'm running out of time. So the one metric that correlated best with insertion tolerance was the radial distance from the central heme axis. It's like a modest correlation, Spearman coefficient of 0.48, but this is pretty striking because it very closely corresponds with the function of MtrA as a protein wire. The iron atoms in the hemes kind of act as like nodes in the path that electrons take as they tunnel through MtrA. And it then makes sense that insertions near the center of this wire are going to be more likely to disrupt electron flow than insertions that are further away. And we think that this result should also probably carry over to electron transfer proteins and other biogeochemically interesting species like Geobacter and other species that use these multiheme cytochromes for extracellular electron transfer. So some takeaways, these cellular assays, growth selections, are really useful for comprehensive mutagenesis and comparison of the function of electron transfer proteins. And this is really great for teasing out sequence structure electron transfer relationships. And after performing this analysis with MtrA, we found that it's surprisingly robust to peptide insertion, although there are some sensitive regions like the heme binding sites and positions close to that central heme axis. Despite this, the robustness of MtrA is really promising for future engineering efforts. We're planning on doing domain insertion with ligand binding domains next to see if we can allosterically regulate electron flow in bioelectronic sensors. So I'd like to acknowledge our funding sources, as well as Joff and Caroline, PIs on the project, my mentor Ian, who's kind of the lead here, as well as Jeff Gralnick at the University of Minnesota for providing the strains. So wrapped up. Any questions?
Nathan.
Great talk.
Thanks.
So the geochemical conditions for dissimilatory ion reduction probably appeared on Earth early in the Archean. But what's interesting is that these extraordinary multi-heme C-type cytochromes are distributed in fairly few microbial taxa. There are, outside the gammas, there are maybe a few delta proteobacteria, and a few firmicutes that have them. So I guess my question is, when do you think these proteins evolved? And why do you think they're so sparsely dispersed in the modern day microbial world?
All right, so Drew, did you have an answer to Nathan's question?
Mm-hmm. Yeah, so I don't know if there's an explanation for that in the literature. I'm not super familiar with it. I do know that like the kind of generally accepted I guess kind of idea for how MtrA evolved is through duplication of other paraplasmic cytochromes like STC and like NERF, I think H. But if I had to take a guess, I would say one is that it's like a pretty metabolically and pretty iron intensive protein to synthesize. So I guess you need to be in like pretty metal rich environments and you need to be able to synthesize all of the hemes, but maybe a more compelling reason might be that like in order to enter the paraplasmic space and insert into MtrA needs to go through like the SEC translocation apparatus, which I would assume probably evolved a little bit later. But yeah, purely speculative.
All right, Drew, thank you very much. Let's give him one more round of applause.